Nucleotides in DNA contains information that is put to work through the production of proteins. Protein synthesis is simply the production of proteins. Proteins form into complex three-dimensional shapes to become key cell structures and regulators of cell functions. Some proteins become important structures in muscle tissues, walls of blood vessels, and transport protein in the membranes. Other proteins, such as enzymes, control chemical reactions that perform key life functions. Transcription takes place in the nucleus. Enzymes make an RNA copy of a portion of DNA strand. RNA is similar but different from DNA. The sugar in DNA is a deoxyribose, while RNA has a ribose. DNA is double-stranded and RNA is single-stranded. DNA and RNA both have 4 nitrogen bases, but RNA replaces the nitrogen base thymine with uracil. There are 3 types of RNA that contributes to the process of making proteins. There is messenger RNA, ribosomal RNA, and transfer RNA. Messenger RNA brings information from the DNA in the nucleus to the cell's cytoplasm. Ribosomal RNA is made up of ribosomes and attaches to the mRNA and uses its information to assemble the amino acids in the correct order. The process of transcription is similar to DNA replication. The process of transcription begins as a copy of gene as mRNA and carries the information to the cytoplasm. The 3 regions in a gene are the promoter, coding region, and termination region. RNA polymerase molecules are present in the nucleus. It is an enzyme responsible for reading DNA code and building the mRNA strand. The promoter signals when to turn the gene on and off. RNA polymerase recognizes when the promoter is signaling to produce the protein and binds to the DNA strand. This causes the double helix to unwind. In the coding region, RNA polymerase reads a single strand and as a result builds the mRNA copy. By reading each nucleotide of DNA, RNA polymerase bonds to the complementary nucleotide, forming the mRNA strand. Then the newly created copy of nucleotides of RNA break free from the DNA, and the DNA goes back to its original double helix form. When RNA polymerase is in the termination region, the sequence causes the mRNA to fold back on itself and prevents transcription from continuing. Both the RNA polymerase and mRNA strand fall off the DNA strand. In summarization, transcription results in an mRNA copy of DNA. There are 20 different amino acids. 3 nucleotides equal 1 amino acid. 1 amino acid equals a codon. So 3 nucleotides equal 1 codon. The order of nitrogen bases in mRNA will determine the type of order of amino acids in a protein. 64 combinations are possible. Translation is the process of converting the information of a sequence of nitrogen bases in mRNA into a sequence of amino acids that make up a protein. Translation takes place at the ribosomes in the cytoplasm. When mRNA arrives in the cytoplasm, ribosomes attach to them and read it. Ribosomes are organelles in the cytoplasm. For proteins to be made, 20 different amino acids dissolved in the cytoplasm are brought to the ribosomes. Transfer RNA takes care of this. On the opposite side of the tRNA molecule from the amino acid attachment site, there is a sequence of 3 nucleotides that are the complement of the nucleotides on the codon. These 3 nucleotides are called the anticodon because they bond to the codon of mRNA. Correct translation of mRNA depends on the joining of mRNA codon with the correct tRNA molecule or anticodon. tRNA is RNA coiled into a certain shape. The nucleotide sequence AUG is the start codon. The ribosome looks for this start codon and begins building the amino acid chain. The ribosome promotes a chemical reaction, causing the amino acids to join by a peptide bond. The ribosome continues down the mRNA strand. The first tRNA releases its amino acid and detaches from the mRNA. This leaves the tRNA molecule free to get and deliver another molecule of its specific amino acid to a ribosome. Again, the ribosome slides down to the next codon and a new tRNA molecule is introduced. Its amino acid bonds to the previous one in the same way. This continues until a stop codon is reached. The newly created amino acid chain folds into a 3-dimensional structure controlled by the order of amino acids. The unique structure gives each protein its own function. During translation, multiple ribosomes read the mRNA strand at the same time, producing many proteins.